Okay, welcome to meiosis. This is after the DNA has condensed into chromosomes, because at first the DNA is chromatin and it's loose, and so this is after it's condensed. So here we have homologous chromosome. These are corresponding from the mom and the dad, and then these are also homologous. And so then during the S phase, if there's enough cyclin, then the chromosomes duplicate, so the DNA is replicated. Okay, so once the S phase has occurred and the G2 phase has occurred, they make sure that the DNA has been replicated correctly, and then the nucleus disappears. So then the chromosome 1 from mom and two from dad come together to form tetrads, just like that. And then they also cross over, which gives genetic variation to the chromosomes. Okay, so next up we have metaphase 1, and this is when the chromosomes line up at the metaphase plate. And another way that there can be genetic variety, I guess, is through independent orientation. So they can line up like this, or like this, or like this. So that's another way that there's genetic variability. And then also the spindle attaches on each side. Okay, so next up we have anaphase 1, in which the bivalents separate, but the sister chromatids don't separate, but they move to the side of the cell opposite. And then also, because the homologous pair splits, then the chromosome number goes from 2n to n, but then there's still 2 chromatids on each chromosome. Okay, so next up is telophase 1, and this means that the daughter cells have 1 chromosome from each pair, and the chromosomes finally reach the opposite pole. And then interkinesis occurs, which creates the furrows and divides the rest of the cell. Okay, so that was the end of meiosis 1. And then, so now we have 2 cells. The nucleus has reappeared, and we enter prophase 2. And there's no more crossing over in prophase 2 because they don't really need to do that, since the chromosomes are no longer homologous, since crossing over occurred in prophase 1, so they don't really need to do that. And then the nucleus disappears once again, and yeah. Okay, so now we have 2 haploid cells, since there's only 2 chromosomes in each cell. At the beginning of meiosis 1, there were originally 4 chromosomes in the cell, so it was a diploid cell, and now it's a haploid cell. There's 2 of them. Okay, so now we have metaphase 2, which the chromosomes line up in the middle. Rather than in metaphase 1, where they lined up in the middle as tetrads, they just line up as individual chromosomes. Okay, so moving on to anaphase 2, the sister chromosomes spread to either the opposite poles, and the spindle apparatus reforms. Okay, so now in telophase 2, the chromosomes decondense, and there's going to be a cleavage furrow in each of the cells. Okay, so after telophase 2 has occurred, and also cytokinesis takes place, we're now going to have 4 haploid daughter cells, and none of them are going to be genetically the same. And yeah, thanks for watching. So in conclusion, the overall goal of meiosis is to produce 4 genetically different haploid daughter cells, and it looks like this run through of meiosis was successful. So there are now 4 haploid daughter cells, and none of them are genetically the same.